So about some proof of concept data that we had actually acquired through a collaboration with Ted Ross when he was at University of Pittsburgh and now has since moved to VGTI in Florida. But I think that the idea behind, demonstrating at least preclinically in the first instance that we could actually cover not only the circulating seasonal epidemic strains, but also potentially protect against all comers, and so therefore really look about protecting against drift, antigenic drift, and even antigenic shift, which was needed, if you can imagine, in 2009 when A California came along and it was a substantial change in antigenic sequence. So we'd really be looking for universal vaccines to cover it to that kind of extent. The other thing is, and this is an adjuvant section which is not lost on me, so this is a presentation that's very much about strategies to induce broad immunity at the level of the antigen itself, but we'll talk a little bit about the role of adjuvants and the need to kind of get that durability of the immune response as well, which will be key. And if you can think about it, in the context of influenza vaccination on an annual basis, typically we do not use an adjuvant, and I think that there is something to be said, well, how do you go from something like that to kind of durable immunity where you probably would be looking at certain types of delivery systems or adjuvants. So just for a little bit of background on top of that, I think that we at Sanofi, and I think at the other major vaccine companies too, would consider it somewhat paradigm shifting to move away from annual vaccination. It would need, as a universal, to cover virus evolution, as I just mentioned. It can have pre-pandemic potential as well, much like in 2009, where you would look to protect against something that would be a major shift from the circulating strains. And even better, if you could move away from the annual vaccination paradigm as well. Now, up until 2009, a lot of work had been done on universal vaccines, but none of them really, frankly, cut the mustard. And that really was, can they protect against disease? Can they replace the traditional seasonal influenza vaccines? And that's what this program hopes to try and do. And so since 2009, and with the introduction of A California, there was the disappearance of the regular seasonal H1 strains as an example. And really, it displaced those, and in fact now that's the prevailing circulating strain. But I think it showed that even when you interrogated immunity in individuals that were either infected or subsequently vaccinated, that there were some key antibodies targeting parts of the hemagglutinin that were quite important and may contribute to universal responses. And we'll talk a little bit about that. But most of the other ones, especially those that are T-cell mediated or matrix protein based, really were not designed to actually replace seasonal vaccines and really would be more of an adjunct positioning used in concert with seasonal vaccines of today. So our program is looking at refocusing universal responses to subtypes specifically, and those subtypes that are circulating today, H1, H3, and influenza B. We do look at pandemic as well, and we look at it in the context of breadth and durability. And we think that today, especially since 2009, that there really is now the offer of some promise to replace strain-specific immunization using inactivated vaccine. So this slide really just talks a little bit about the protection against disease issue in as far as which antigens would you pick if you wanted to replace seasonal influenza vaccines of today. And we've kind of looked at our antigen list, if you like, here in the context of something that would either prevent disease, which really relies upon the hemagglutinin as the main component of the vaccine. It is also the main component of influenza vaccines of today. And separating it from more the matrix-based proteins or nuclear protein, which really either only satisfy protection against influenza A strains and not necessarily both A and B, but also they really do not prevent infection. They're really designed to modulate outcome of disease. So for us, our program rests on the full-length hemagglutinin and how we might harness that to induce universal responses. if you look at this from a target product profile perspective, and what's important is breadth, and as you go along this axis, covering all types, and so therefore gaining more breadth versus durability. You can see that if in this corner here we represent standard of care, which is a matched vaccine, you can see that it's very strain specific, it's used annually and has little breadth. And what we're hoping to do, and what we're developing, are antigen and antigen compositions. This one is without an adjuvant, frankly, that would satisfy breadth and protection against all circulating strains and even mismatched strains, versus ones that in fact are designed to last a lot longer, moving away from annual vaccination through the addition of an adjuvant. And we actually do this separate from vaccines that would target pandemic strains or have as a pre-pandemic vaccine, which could be added to the mix if needed. So the program hypothesis really is centered on universal vaccines being rationally engineered to elicit broadly cross-neutralizing antibodies. So we're talking about refocusing immunity, especially in the context of people that have already been pre-vaccinated, obviously, which really is the target population, one target population, in as far as that they're either vaccinated every year or in fact people that have seen natural infection. But we really are talking about recalling the right type of immunity through re-engineering the vaccine candidates themselves. And I think it's an interplay between antigen selection, the delivery, and the formulation itself, which really will get you there. So hemagglutinin-based universal influenza vaccines really is reliant upon conserved sequences. You have here the trimeric HA, post-2009. There was a lot of people, whether it be Gary Nabel out of the Vaccine Research Center at NIH, or whether it be Peter Palese at Mount Sinai, really a lot of people were focusing in on the stem and broadly cross-neutralizing antibodies directed against the stem region of the hemagglutinin. We have been focusing more on the head, maybe looking to combine that with responses to stem-based immunity, but with collaborations either with Ted Ross at VGTI or through our collaborations in China at Xiamen University, we found that through sequential immunization or infection, you can actually generate broadly cross-neutralizing antibodies at the level of the head, which are functional and that work by HAI. And these are some of the benefits of generating an immune response against the head that is designed to prevent against infection, which this one here may not be. Problem is that when you work with the head, it's more subject to antigenic variation, and that's something that we have to take on board. So I just listed here, not to go through everything, but some of the preclinical proof of concept studies that we would like to see as part of our vaccine program, and as it relates to kind of translational research activities that we would want to do. But we need to be able to demonstrate in our program that we have a full-length HA that is structurally sound. If we're going to test it, and we hope to do so even as part of inactivated vaccine formulations, know that we can rescue it as live virus. It's got to be recognized by very interesting antibodies, ones that usually give you breadth by neutralization. We need to demonstrate that breadth not only immunologically against strains from multiple years, but we need to show efficacy both homologous and heterologous. It's got to be non-inferior to standard of care, which is the matched inactivated vaccine, but it has to be superior in its ability to protect against a mismatched strain, and so I'll show you some proof of concept data there. And I think what we're very interested in looking at is this, just the hypothesis that says that we can recall back immunity for people that have actually seen influenza, whether it be vaccine or infection, but those really important antibodies that they've already seen. And so we do a lot of work in pre-existing immunity models, in ferrets especially, to try and validate that hypothesis. And we also do that at the level of studying vaccination in humans as well. And I won't talk too much about that today, actually. The durability piece is going to be important from the perspective of obviously moving away from annual vaccination. Oh, All right, so this was the proof of concept data with H5 using an approach called Cobra, which is computationally optimized broadly reactive antigen. It basically used this layered consensus-based approach to generate a consensus HA full-length sequence. When you look at it from a percent identity perspective up against, now in the context of H5 strains belonging to different clades, the total HA identity is very strong with Cobra-based HA designs, but antigenically, it's very different. If you look at a dendrogram here of the different clades of hemagglutinin, if you look at the clades and the sequences within those clades of viruses or HAs of viruses from H5, antigenically they're quite distinct. And what we're doing here with a Cobra is trying to find one that would basically neutralize each one of these viruses within these clades. This was some of the initial data that showed, well, first of all, the methodology around developing consensus-based designs, layering them so you would have a secondary consensus and then ultimately a final consensus sequence that when put into mice, and in this instance it was a couple of doses. Early studies always used alum in order to get kind of that bolstering of the immune response. But what you see are VLP-based immunogens, I should have stated that before, sorry, that was with neuraminidase and M1, that when given as 2 doses with alum, induced goodly antibody responses, this is total IG, that recognized strains from each of the clades and was as good as wild-type or homologous sequences. But when you looked at functional antibody responses, what you can see is that a VLP, which is targeted and strain-specific, was inferior even to the consensus-based design, which actually was able to neutralize or exhibit HAI activity against all of the clades that were evaluated. And this was evaluated in mice, it was evaluated in ferrets, then it went into non-human primates, and we saw basically exactly the same thing, where in fact you can see that the VLPs that are consensus-based designs, when given with an adjuvant, actually affords HAI responses across many, many strains, across all of the clades and subclades of H5. And in fact, from a histopathology perspective, whether it be, you can see here, animals that were given Cobras did not suffer with an inflammation, much like the uninfected controls, whereas the whole cell inactivated vaccine did result in an inflammation in the lungs. So this was kind of a very nice profile from the perspective of a consensus-based design, giving minimal inflammation associated with immunization and then challenge. This went then on to develop second-generation vaccines, and you can see here, now if this is the full-length hemagglutinin for H5, and now we've separated it into identifying the head and the stem, you can see the subtle changes that are happening as we build newer designs that are really either designed to incorporate, say, maybe avian sequences as an example, or just build up a much deeper database of sequences and build new antigens. But this one, similarly, when looking at human avian cobras or an all H5 cobra, was similarly evaluated now in ferrets, and you can see that the human avian cobras were actually also very good at inducing functional antibody responses against all of the strains within these clades that are evaluated here. And what we did is we took these, put them into chickens as part of this second generation H5N1 Cobras, 2 doses again. This time we're using montanide as an adjuvant to get the level of antibody responses high and hopefully durable. And then we did 2 challenge studies, one which was representative of a homologous challenge and one which was heterologous. So we took the human avian cobras or the fully human cobra, and we took whoopaswan, which was basically matched to the challenge strain here as a VLP or as a killed vaccine, and against something which was still virulent, but not quite as virulent as this heterologous challenge, everybody was protected. when you can see over here, animals then challenged with, or another group of animals challenged with a hotter, more virulent heterologous strain, that in fact the cobra, in fact the all-human cobra, in fact, surprisingly enough, actually provided much better efficacy profile from a survival perspective than even matched inactivated vaccine. So you can see that from a proof of concept standpoint, using H5, the ability to generate broad neutralizing antibody responses, functional antibody responses, and get protection against homologous and heterologous challenges is what we were looking for. And this is why we use this as to set us up for the work that we wanted to do in seasonal influenza vaccine studies. Because you can see that in the context of H5, the number of sequences that you're looking to cover, while large by number of subclades, by number of H1 sequences we're looking to try and cover for a subtype universal, that's quite small, and our task is therefore much harder. So we used a similar strategy. It's all about the full-length HA. It's about modifying the head and validating it through recognition with key neutralizing antibodies and identifying those clusters of H1 sequences and then prototypic viruses that we can use, usually vaccinal strains, to kind of document, are we able to protect against all of these H1 vaccinal strains as a sign of how well we're doing in developing an H1 universal. And this is what we're looking to do as a proof of concept for all of the seasonal viruses, H3 and influenza B as well.
Can we start to wrap up?
Yeah, sorry. So this just goes to show a little bit more detail about those viruses that we're looking to protect against and how, in fact, the Cobras that are designed, where they fall on a kind of a PCA plot, it's another version of a dendrogram. And what we're looking at here is those new consensus-based designs and how they react with either stem or broadly neutralizing head antibodies and what years they're designed to cover. And in fact, when you look at that now in mice, and we're getting close to the end, what you can see is the functional antibody response by HAI induced by inactivated vaccine for A. California. You can see that it's predominantly generating functional antibody responses against itself and nothing else for these viruses that cover, at least in this region here, around about 30 years. But Cobras, either P1 or X6, you can see are giving us neutralizing functional antibody responses against more viruses. We can cover not only A. California and that 2009 pandemic strain, but also earlier viruses too. And if you combine it with X6, which we think this could be a potential cocktail of two antigen designs to cover 30 years or so of viruses for H1. And they have good morbidity profiles. And again, you can see these are with subtle changes and tinkering in the head that distinguish, something like P1 and X6 from the wild-type virus itself. And so just in summary, we think that Cobras themselves as an approach and the idea of re-engineering hemagglutinin at the level of the head and around the receptor binding site is as a way of demonstrating that you can get breadth of neutralization. We can get 30 years coverage just based on the preclinical efficacy studies that we've looked at. And it's matched to standard of care. It covers us for drifted and mismatched strains, at least in the context of H1. But I think the big thing is about the immunogenicity and efficacy in pre-immune animals, which we've started to do, where the levels of antibody responses are even higher. And that will maybe be important, especially when considering durability. And that's where we need to go to get away from the annual vaccination paradigm. And these are really some of our next steps. So with that, I'll just thank our collaborators and acknowledge the folks in Sanofi working on this program, similarly at VGTI Florida, the folks at NIDVD at Shannon University for the antibodies, and also people involved with the chicken study, including Mary Ellen USDA. Okay, that's it.